Question 1. Which statement best describes how enzymes affect chemical reactions? A. They change delta G to make endergonic reactions spontaneous. B. They lower activation energy and speed up forward and reverse reactions without changing delta G or the equilibrium constant. C. They raise activation energy to control rates. D. They shift equilibrium toward products. Correct answer B. Enzymes lower the activation energy, increasing rate in both directions, but they do not alter delta G or the equilibrium position. Question 2. How does temperature typically influence enzyme activity? A. Activity always decreases as temperature rises. B. Activity rises with temperature to an optimal point, then declines as the enzyme denatures. C. Temperature has no effect. D. Cold permanently denatures most enzymes. Correct answer B. Increased kinetic energy speeds collisions up to an optimum. Further heating disrupts structure and reduces activity. Question 3. In competitive inhibition, how are enzyme kinetics affected? A. Km increases while Vmax is unchanged. B. Km decreases while Vmax is unchanged. C. Km is unchanged while Vmax decreases. D. Both Km and Vmax decrease. Correct answer A. A competitive inhibitor competes at the active site, raising apparent Km. Sufficient substrate restores Vmax. Question 4. Feedback inhibition most often works when the pathway's end product, A, binds the active site of the first enzyme to speed it up. B, binds an allosteric site on an early enzyme and decreases its activity. C, binds to DNA to increase transcription. D, binds ribosomes to halt translation. Correct answer B. The end product allosterically inhibits an earlier step, preventing unnecessary accumulation and conserving resources. Question 5. What is the role of coenzymes such as NAD+ and FAD in metabolism? A. Structural parts of the active site only. B. Allosteric inhibitors. C. Electron carriers that accept and donate electrons during redox reactions. D. Primary cellular energy currency like ATP. Correct answer C. Coenzymes shuttle electrons and protons between reactions, linking oxidation and reduction steps. Question 6. Where does glycolysis occur in eukaryotic cells? A. Mitochondrial matrix. B. Cytosol. C. Inner mitochondrial membrane. D. Peroxisome. Correct answer B. Glycolysis takes place in the cytosol and yields pyruvate, ATP, and NADH per glucose. Question 7. What are the net products of glycolysis per glucose? A. 4 ATP, 4 NADH, 2 pyruvate. B. 2 ATP, 2 NADH, 2 pyruvate. C. 2 ATP, 4 NADH, 2 acetyl-CoA. D. 0 ATP, 2 NADH, 2 pyruvate. Correct answer B. Glycolysis nets 2 ATP, 2 NADH, and 2 pyruvate molecules per glucose. Question 8. In eukaryotes, pyruvate oxidation to acetyl-CoA occurs where and produces what? A. Cytosol, ATP only. B. Mitochondrial matrix, acetyl-CoA, NADH, and CO2. C. Thylakoid lumen, NADPH. D. Nucleus, FADH2 only. Correct answer B. Pyruvate dehydrogenase in the mitochondrial matrix produces acetyl-CoA, NADH, and carbon dioxide. Question 9. Per acetyl-CoA, the Krebs cycle directly yields A. 3 NADH, 1 FADH2, 1 ATP or GTP, and 2 CO2. B. 2 NADH, 2 ATP, 2 CO2. C. 1 NADPH, 1 ATP, 1 CO2. D. Only ATP. Correct answer. A. Each turn of the cycle generates 3 NADH, 1 FADH2, 1 ATP or GTP, and releases 2 molecules of CO2. Question 10. The immediate role of oxygen in cellular respiration is to A. Accept electrons and protons to form water at the end of the electron transport chain. B. Split glucose into pyruvate. C. Pump protons across the inner membrane. D. Act as a coenzyme for ATP synthase. Correct answer A. Oxygen is the terminal electron acceptor in the ETC, forming water when reduced. Question 11. During oxidative phosphorylation in mitochondria, the proton concentration is highest in the A. Mitochondrial matrix, B. Intermembrane space, C. Cytosol, D. Nucleus. Correct answer B. The ETC pumps protons from the matrix to the intermembrane space, creating a gradient used by ATP synthase. Question 12. Chemiosmosis refers to A. ATP production by substrate-level phosphorylation. B. Proton diffusion through ATP synthase driving phosphorylation of ADP. C. Active transport of glucose into cells. D. Electron transfer between NADH and FADH2. Correct answer B. The proton motive force powers ATP synthase to convert ADP and inorganic phosphate into ATP. Question 13. The primary purpose of fermentation is to A. Produce large amounts of ATP, B. Generate oxygen, C. Regenerate NAD+, so glycolysis can continue when oxygen is limited, D. Produce acetyl-CoA. Correct answer C. Fermentation pathways recycle NADH to NAD+, allowing glycolysis to produce ATP anaerobically. Question 14. The light reactions of photosynthesis occur where and produce what? A. Stroma, glucose only. B. Thylakoid membranes, ATP, NADPH, and O2. C. Matrix, ATP and CO2. D. Cytosol, NADH. Correct answer B. Light reactions in thylakoid membranes split water, release oxygen, and generate ATP and NADPH. Question 15. The Calgon cycle primarily A. Uses light energy to split water. B. Fixes CO2 into organic molecules using ATP and NADPH to produce G3P. C. Produces oxygen. D. Oxidizes pyruvate to acetyl-CoA. Correct answer B. In the stroma, rubisco fixes CO2, and the cycle reduces it to glyceraldehyde 3-phosphate using ATP and NADPH. Question 16. Which statement best describes the relationship between the light reactions and the Calvin cycle? A. They are unrelated. B. The Calvin cycle directly requires light photons. C. ATP and NADPH from the light reactions power carbon fixation in the Kelvin cycle. D. The light reactions consume CO2. Correct answer, C. The light reactions supply chemical energy and reducing power for CO2 fixation in the Kelvin cycle. Question 17. Compared with non-cyclic electron flow, cyclic photophosphorylation, A. Produces both ATP and NADPH. B. Produces ATP only and no oxygen. C. Produces NADPH only. D. Produces oxygen only. Correct answer, B. Electrons cycle around photosystem 1 to pump protons and make ATP without making NADPH or O2. Question 18. When stomata close during hot, dry conditions in C3 plants, what is the immediate effect on photosynthesis? A. Internal CO2 rises, increasing Kelvin cycle rate. B. Internal CO2 falls and O2 rises, promoting photorespiration and reducing sugar production. C. Light reactions stop completely. D. Chlorophyll is irreversibly destroyed. Correct answer, B. CO2 scarcity and elevated O2 favor rubiscose oxygenase activity, decreasing net carbon fixation. Question 19. How does ATP hydrolysis drive endergonic reactions in cells? A. By lowering activation energy alone. B. By providing heat. C. By transferring a phosphate group to intermediates, shifting their delta G and coupling the reactions. D. By directly donating electrons to substrates. Correct answer, C. Phosphorylation changes intermediate free energies and couples exergonic ATP hydrolysis to endergonic steps. Question 20. Which statement about Gibbs free energy is correct? A. Exergonic reactions have positive delta G. B. Endergonic reactions have negative delta G. C. Exergonic reactions have negative delta G and can proceed spontaneously. D. Delta G is unaffected by concentration. Correct answer, C. A negative delta G indicates a thermodynamically favorable process, though the rate may still require catalysis. Question 21. At chemical equilibrium, which is true? A. Delta G equals 0 and there is no net change in concentrations. B. Delta G is highly negative. C. All reactants are consumed. D. Enzymes are inactive. Correct answer, A. Forward and reverse rates are equal. Delta G is 0, though molecules continue to interconvert. Question 22. An enzyme-catalyzed reaction is at saturating substrate concentrations. Which change best increases the reaction rate? A. Add more substrate. B. Lower the temperature. C. Add more enzyme. D. Add a competitive inhibitor. Correct answer, C. At saturation, Vmax depends on enzyme concentration. Adding enzyme increases maximum rate. Question 23. Allosteric activators generally: A. Block the active site. B. Stabilize the high-affinity conformation of the enzyme, increasing activity. C. Denature the enzyme. D. Convert endergonic reactions into exergonic reactions by themselves. Correct answer, B. Activators bind sites distinct from the active site and shift the enzyme toward a more active state. Question 24. What is the effect of an uncoupler that dissipates the proton gradient in mitochondria? A. ETC and ATP synthesis both increase. B. ETC slows and ATP synthesis increases. C. ETC may continue or increase while ATP synthesis drops sharply. Energy is released as heat. D. No change occurs. Correct answer, C. Collapsing the gradient lets electron transport proceed without efficient ATP production, generating heat instead. Question 25. Under high light but low CO2, which factor most limits photosynthetic sugar production in a C3 leaf? A. Light intensity. B. CO2 availability for the Kelvin cycle. C. Water splitting in photosystem 2. D. ATP synthase activity only. Correct answer B. With abundant light, the Kelvin cycle becomes CO2 limited. Low CO2 restricts carbon fixation despite ample ATP and NADPH.